Good morning to you all. I am Dr. Thenmozhi, Assistant Professor and Head, Department of Zoology, St.Joseph's College of Arts and Science, Autonomous Cuddalore. I am going to talk about transgenic plant and transgenic animal. Transgenic plant is a modified organism where genes are transferred from one organism to another organism through genetic engineering techniques. What is its purpose? Why do we want a transgenic plant? Lot of advantages are there. Mainly the purpose of producing transgenic plant is to obtain species that has the ideal traits, high yield and quality. The two important methods used for gene transfer are agrobacterium mediated gene transfer, particle bombardment or gene gun method are the two methods used for gene transfer. Actually, by gene transferring only we can produce new type of plants and animals. First method, agrobacterium mediates is gene transfer includes agrobacterium. From the name itself you can see it is a bacterium. Its full name, scientific name is Agrobacterium tumefaciens. It is actually a plant pathogen. You know, pathogen means disease causing organism. So Agrobacterium tumefaciens is a bacterium which causes disease in plant. What disease it causes? It is known to cause crown gall disease, which is swelling in plants just above the soil level. After infecting the plants, they transfer their genetic material to them, which eventually gets incorporated into the plant gene. So after infecting a plant, they usually the bacterium or a virus, after entering into a cell, they will transfer their genetic material, they will incorporate their genetic material into the cell itself. What cell it is affecting, that cell itself it will send its genome. Obviously they gets incorporated into the plant genome itself. For genetic engineering, bacterium incorporated with the TI plasmid with the desirable genes are made to infect the plant. For genetic engineering, we are using some of the desirable genes from a bacterium. Usually the desirable gene is from TI plasmid. You know very well about plasmid. Plasmid is an extra genome that is present in the bacterium. Apart from their normal DNA, normal genome. So it is a ring type DNA which is called as plasmid, which is usually used in genetic engineering. They are considered as a desirable genes and made to infect the plant. First of all, the desirable gene we wanted to send is, we have to take it from a bacterium, that is TI plasmid. That genome is taken from TI plasmid. The TI plasmid is a tumor indicating circular plasmid. As I said already, it is a circular plasmid that transfer the host chromosomes to the plants and also responsible for causing a swelling disease. It is a gall disease. Gall means swelling disease. It will cause a swelling in the plant. So by this method, we can use the gene transfer from the desirable gene containing bacteria into the plant itself. Then, second method is particle bombardment or gene gun method. As the name suggests itself, this is the desirable gene is coated with the gold or tungsten particle. So, just like a gun, sending a gun into just like a gun, it will be bombarded into the plant cell itself. Once bombarded, the sequence is incorporated into the plant cell itself. Once it bombarded, automatically it will be incorporated into the plant genome itself. So which can be proliferated by the tissue culture method. Then one cell can be proliferated into millions of cells by tissue culture method, followed by a tissue culture method. What are all the applications of plants? Transgenic plants, not normal plant, transgenic, transferring up of new genes into the plants. First application is resistant to biotic and abiotic stress. What is meant by stress? Stress means something causes depression. Biotic stress means imposed on plants as a result of action of living beings such as viruses, bacteria, pests and other pathogens. Biotic means living organism. Because of the living organisms, there are some stress caused in the plants. That is what called as biotic stress. Abiotic stress means non-living things, anything that can be a pesticide, that can be a fertilizer, that will cause some stress in the plant. Just like that, first we shall see about abiotic stress. First we can see biotic stress, that is as a result of attacked by viruses, bacteria, pests or pathogens, the plants will get some biotic stresses. In order to relieve the plants from such biotic stress, they are incorporated with the disease resistant genes, which gives a better yield and quality to the crop. What is abiotic stress? This is also one of the application of transgenic plants. Transgenic plants could not get any abiotic stress because of the desirable gene transferred in it. Abiotic stress is as a result of changes in the environment causes great damage to the plants. For example, soil composition, humidity, water level, temperature are some of the important factors for plant growth, without which they cannot grow. So that due to the changes in the climate, all the factors seems to be altered. Thus the plants are incorporated with the stress tolerant genes for better production. This is one of the important method of producing transgenic plants, so that due to the changes in the climate or any other non-living factors, that will not affect the plant, because the plant is having all those resistance in their genome itself. Next important application is increased nutritional value. Biofortification is one of the term that is used. That is, it is a process of increasing nutritional value of the crop. Nutritional value increasing is one of the important thing that can be applied in the modern biotechnology, in which increasing the nutritional value of the crop is one of the essential factors that is implemented in the production of transgenic plants. Okay, so the transgenic plants will give better essential nutrients in it. Malnutrition is a common factor you can see everywhere in developing countries. So even in developing countries also, malnutrition is one of the very important bad factor which suppresses the human resources. So in order to give a solution for this, plants are engineered to produce crops of better nutritional value. So which is possible only by producing transgenic plants. Factories for production of recombinant proteins. Recombinant protein production, in that also it is one of the important application in it. Recombinant human proteins have been produced using animal and microorganism system. But due to some shortcomings, it has been shifted to plant system. Vaccines and antibiotics have been obtained from transgenic plants. Lot of vaccines, lot of antibiotics have been we can obtaining from transgenic plants only. So this application is still in the development stage only and have not been commercialized yet. Examples of transgenic plants. Golden rice is one of the rice which is produced to overcome the deficiency of vitamin A. Vitamin A deficiency is a very common thing because of which people are suffering with eye problems, eye irritation, inflammation in the eyes, or because of vitamin A. So in children especially, so using the gene gun method, the rice species are incorporated with the phytoene synthase genes, which increases the vitamin A. So in order to increase the vitamin A in the rice, the gene which is essential for producing vitamin A, that is called as phytoin synthase genes. Okay, so that will be incorporated into the rice grains. Next example is BT cotton. BT cotton is genetically modified crop. Bacillus thuringiensis is a bacterium which produces a disease in the caterpillar that affect the cotton balls. So BT cotton is a plant, the cotton plant, which is modified with this Bacillus thuringiensis genome, that is the bacterium which causes disease against the pest of cotton. Then flavor saver. Flavor saver is a genetically modified tomato crop that has longer shelf life. Usually the tomato will decay very easily because its skin is very soft. So in order to avoid that, to keep the tomato for a long time without decaying, so flavor saver is a genetically modified tomato crop. So that is due to that will causes what? Delays in ripening and softening. So several other plants like potato, corn, papaya, maize, squash, pumpkin, alfalfa have been modified to obtain crops with better yield and resistance. What are transgenic animals? Transgenic animals are the animals with the modified genome. A foreign gene is inserted into the genome of the animal to alter the DNA. This method is done to improve the genetic traits of target animal. Initially, the improvement of genetic traits was done by selecting breeding methods. The animals with the desired genetic characteristics were mated to produce an individual with the improved genetical characters. Since this technique is time consuming and expensive, it was later replaced by recombinant DNA technology, is a modern technology. Transgenesis is a phenomenon in which the foreign gene with the desired characters is introduced into the genome of the target animal. The foreign gene that is introduced is known as transgene, and animal whose genome is altered, and then it is known as transgenic. These genes are passed into the successive generations. Transgenic animals are generally engineered and are known as genetically modified organisms. Animals with the transgenic characteristics are known as genetically modified organism. First genetically modified organism was engineered in the year 1980. So by some of the methods like 4 methods. Physical transfection. In this method, gene of interest is directly injected into the pronucleus of the fertilized ovum. It was a very first method that is proved to be effective in mammals. So chemical transfection is another method of gene transfection, include transformation. In this method, target DNA is taken up in the presence of calcium phosphate, and the DNA and the calcium phosphate co-precipitates that facilitates the DNA uptake. Mammalian cells possess the ability to take up the foreign DNA from the culture medium. Retrovirus mediated gene transfer is another method, and this gene is transferred by means of a vector. Since retrovirus have the ability to infect the host cell, they are used as a vectors to transfect the gene of interest into target genome. Viral vectors. Vectors used to transfect rDNA into the animal cell. The viruses possess the ability to infect the host cell and expresses well and replicate efficiently. Bacterial infection is another method by which gene of interest is transferred into the target gene with the help of a bacteria. Dolly sheep is the first mammal that is cloned from the adult cell. So in this method, the udder cells from 6-year-old Finn desert white sheep were injected into an unfertilized egg from Scottish blackface eve, which had its nucleus removed. The cell was made to fuse by electrical pulses. After the fusion of the nucleus of the cell with the egg, the resultant embryo was cultured for 6 to 7 days. It was then implanted into another Scottish blackface eve, which gave birth to transgenic sheep doll. Transgenic mice is another example for transgenic animal. So in this you can see the applications of transgenic animals finally, that is normal physiology and development, that is growth factor can be altered in the transgenic animals by the study of gene regulations. Study of diseases. Transgenic animals can be used for research in the development of medicines. For example, transgenic models for diseases such as Alzheimer's and cancer diseases. Alzheimer's and cancer diseases are because of it has to be studied by transgenic animals. So biological products, then vaccine safety or other applications involved in transgenic animals.